Hello, good morning, good afternoon. Thanks for joining us in this second presentation for the International Plant Breeding Seminar Series. We are in the ninth season, and please tell us from where you're joining, and we are very happy to have you here. We're also very happy to have Dr. Paula Silva from Uruguay, who's going to be presenting about pre-breeding in action. Paula has earned her doctorate from Kansas State University in the area of genetics. Her area of studies focuses on crop genetics and genomics with emphasis on breeding for disease resistance using advanced phenotyping and genotyping tools. Paula is also interested in exploring new sources of resistance through the use of wild species. She enjoys learning and applying new insights in genomics and breeding, and equipping herself with the knowledge and experience needed to tackle future challenges and innovative ideas and rigorous scientific perspective. Paula, it's a pleasure to have you with us. It's a pleasure to meet with you again. And please take it from there. I'm going to stop sharing. Yeah.
All right. Thank you, Carlos. Thank you for the introduction and for the invitation to present in this seminar series. It's a real honor for me. We met with Carlos back in 2017 when
something like that.
Yeah, when I was doing my PhD in Kansas. So he is a very good friend of mine. So thank you for the invitation.
You're welcome.
So I'll share my screen. Here we are. All right.
You're good. You're good. Yes, you're very good.
All right.
Go ahead.
Okay, so today I will be talking a little bit about the research activities I'm doing here at INEA, and most of them are related to pre-breeding for wheat and barley. So back in 2019, when I was still at K State, I was appointed to be the pre-breeder lead of wheat and barley, mainly wheat and barley. And so our team focuses on different things, but mostly we do breeding for disease resistance. We also do a little bit of pathology for the cereal rusts, doing a little bit of epidemiology. Most of our work is applying field-based phenotyping. And more recently, we started to work with breeding for physiological traits and a little bit for quality traits. And we also have some participation on applied quantitative statistics. Today, my presentation will be focused on breeding for disease resistance. And I mean, it's the main thing I do for research, but I like to talk about this because breeding for disease resistance is a real challenge. I mean, we have so many pests and diseases that affect the crops, and each of these diseases has a different importance in different regions, right? And so one of the things that is important to remember is that the pathogens are evolving, constantly evolving. And even if we develop a resistant variety today, there is no guarantee it will stay resistant tomorrow. Although the plants have developed a mechanism to resist the diseases, the nature of the host pathogen interaction means that they are all the time emerging and overcoming the resistance. And so new virulent strains can emerge and overcome the resistance, and this is like an ongoing race. Second is because breeding for disease resistance is a continuous process that requires some strategic management. So this means we cannot deploy resistant genes individually. It's ideally to deploy them in combinations. And for this, we need to monitor the pathogen population constantly, because if we don't handle this properly, the risk of losing, your effective resistance is imminent. And then the third key aspect is that the breeding programs are constantly screening for diverse germplasm. It could be wild relatives, or it could be land races, or even elite lines, to search for these new sources of resistance. And this is essential to diversify the genetic base and build more durable resistance. So 3 key aspects to consider when doing breeding for disease resistance is knowing the pathogen variability, knowing the genetic diversity you have in the host, and also the development of resistant germplasm. So I will talk about these 3 aspects using the wheat rust as a model pathosystem. So the wheat rusts are an excellent example to talk about breeding for disease resistance. So we have 3 major wheat rusts. On the left, we have the stripe rust that is caused by Puccinia striformis tridici, and it drives on cooler conditions, typically around 10 and 15 degrees. Then we have in the center the leaf rust caused by Puccinia triticina that prefers moderate temperatures around 20%, and is the most widespread globally rust of the 3. And then on the right, we have a stem rust that is caused by Puccinia graminis tridici and develops at warmer temperatures. And I think it's the most feared of the 3 of them. So at the bottom, I'm highlighting how many resistant genes have been named for these 3 rusts to date. So 87 genes for stripe rust, 85 genes for leaf rust, and 66 genes for stem rust. And so starting with the process of how do we identify the races, so we need to study the pathogen variability. And one of one thing we can do is to study the pathogen variability. So for these, we do annual nationwide surveys to collect samples. So here you have a map on the bottom that shows all the sampling points, around 250 sampling points in Uruguay in 2021, that we just go to barley and wheat fields and we sample the rust, and then we took it to the greenhouse. And in the greenhouse, we use differential line sets. This is a group of isogenic lines, each of them carrying a known resistant genes. And we use these lines to inoculate with the sample that we brought from the field. And then using a visual scale, we either say that line is susceptible or resistant, and we use a pattern of response across these differential lines to come up with a letter. And at the end, we have a code for that race, for that sample that we took from the field. So in this example, the race we analyzed was grouped to the TDT1020 race, okay? So this system helps us to track how the rust population is over time and space. And of course, it's something that we use to inform breeding programs. So my first example here is for wheat stripe rust, and it's part of Venancio's PhD thesis. So one of his chapters is about identifying the genetic groups present in Uruguay. So a little bit of the background story here. Stripe rust was not a problem until 2016 in Uruguay, but suddenly in 2017, it became a problem. So here you can see in the map the surveys that we do annually. And you can see in 2016, all the samples were free of stripe rust, but in 2017, many of the fields that we sampled were showing symptoms. And we said, well, what happened here? I mean, something, must be happening in the pathogen population. And so we knew in 2015, some countries in South America were having some introductions from Europe. And so Venancio was, what he did, he took samples from 2017 through 2021, and he went to do an internship at Denmark at Aarhus University and the Global Rust Reference Center. And he did phenotypic analysis and genotypic analysis. And he came up to that the Uruguayan population of stripe rust is composed with 3 genetic groups, PSTS 7, 10, and 13. And within the 13, we have 3 variants that they are independent mutations on 3 different genes. So, with his study, we kind of now know what is happening in the population. We know that the population is evolving, but most importantly, that is rapidly evolving. And that is something that we use for all the rust to study how is the pathogen population changing over time. And so this type of a study is giving us very valuable insight into how is the rust pathogen population, because allow us to precisely identify what are the races that are present in the country. But more importantly is because the global rust research community is using the same sets and the same scoring system. That means, we are all speaking same language, kind of. Thanks to that, we can compare our findings to the findings from other countries, and we could track the spread of the virulence across some regions. So we know, for example, if something appears in Europe or in Brazil, then we know if that race is most probably to arrive to Uruguay a few years later, right? And so here I want to also highlight Silvia Germán, because most of this work has been done under her leadership. She is the former researcher doing this type of a study. So a lot of this is based on her scientific work. So I want to thank her. And doing this, we know exactly how is the population for the stripe rust, the leaf rust, and the stem rust. And many different publications came up out of this work. Another way to study pathogen variability is using genotypic data. It gives you a deep insight into the pathogen diversity. So here I'm showing 2 examples that how we have used this approach. So one is in collaboration with Jim Scalmer's group from the University of Minnesota. So he was analyzing the wheat leaf rust population using GVS, genotyping by sequencing. And, these allow to identify the global genetic groups of Puccinia triticina around the world. And because we submitted samples from Uruguay, we know exactly what is happening in Uruguay compared to the rest of the world. The other example on the right is a collaboration we have with Melania Figueroa's group from the CSIRO in Australia. And they have been working on stem rust using whole genome sequencing. So this work is helping us understand how new virulence are arising for stem rust. And, we know exactly how is the population from Uruguay looking. So we know that we have 5 different phylogenetic groups. Each of them has very distinct characteristics. So together, these studies are showing us how the genotypic tools complement the phenotypic data and how powerful they are, to track the pathogen population. So the second aspect that I want to talk about is genetic diversity in the host. So this is very important to understand, not only like to combine with the pathogen population variability, but also to do some like basic pre-breeding work. And so when it comes to resistance, we can classify it in 2 main types. One is the qualitative resistance, and the other one is the quantitative resistance. And so let's walk through the differences of these 2 types. So if we talk about at the phenotypic level, we have the qualitative resistance, has 2 distinct classes, resistant or susceptible, like a switch, and, like an on-off switch. And then we have the quantitative resistance that is more like a continuous distribution of disease severity, like a gradient of responses across the population. Then at the population level, the qualitative resistance is often like a 3-1 segregation ratio, like a classic Mendelian inheritance. And then for the quantitative resistance, this involves like multiple genes of small effects, kind of like showing like a continuous distribution and a more complex inheritance. At the genetic level, we have for the qualitative resistance, we have like a single gene, clearly distinct phenotypes. And on the quantitative side, we have a single gene with a small effect, and the final phenotype depends on the combination of many of these genes, right? So understanding these differences is essential to design effective breeding strategies, okay? So again, I will just, what we just discussed, put it in on perspective of the wheat rust. So we have the quantitative versus the qualitative versus the quantitative resistance. And so on the left, we have the qualitative that is called race-specific resistance. And it's a type of resistance that triggers a hypersensitive reaction. That is when the host kills the infected cells around the infection site, so the pathogen cannot spread. And this type of resistance is often very effective, but is short-lived. This is because the pathogen can overcome quickly, because a single mutation in one gene is enough to overcome this resistance. And it's mostly expressed, it's expressed at both seedling and adult plant stages. So that's why it's also known all-stage resistant, because it works all the time. On the other side, we have the quantitative resistance that is also known as non-race-specific resistance, because it is not dependent on the race of the pathogen, doesn't trigger the hypersensitive reaction, and is typically more durable over time, because it allows the pathogen to grow, so the pathogen doesn't need the pressure to mutate. It can develop on the host. And so this resistance is it usually kicks only at the adult plant stage. That's why it's also known as adult plant resistance, okay? And so one of the activities our groups is doing is on the genetic diversity in the host, is phenotypic characterization and genotypic characterization. So on the phenotypic side, we establish field nurseries specifically designed for each disease. So we have one disease nursery for striped rust and one for leaf rust and one for stem rust. Each of those are planted in different dates to, favor the disease that we are looking for. And we typically work with 1 meter row plots and spread the rows that we artificially inoculate with the races that we know we have present in the country, right? And so most of the time we evaluate introductions in cultivars and advanced breeding lines. We also offer phenotyping services for other institutes because this, is a very beautiful setup for screening material. But sometimes we cannot screen for some of the races or for some diseases. So in that sense, we also send material to evaluate to another countries or another regions. So in this example here, I'm showing how we sent every year a group of wheat lines to Kenya to characterize against the UG99 race complex for stem rust, because we don't have it here in Uruguay, but they have it there. So, using this type of information, we know, for example, wheat lines that are resistant to the UG99, but most importantly, we know about lines that are resistant in Uruguay, but are not resistant to UG99. So kind of give us information, to be prepared if something, if the UG99 arises in the country. And then on the genotypic side, we use molecular markers associated with genes, known genes, to characterize materials. So in this example, I'm showing the CASP genotyping, but we also use other type of markers like SSRs or CAPs. So to better characterize the lines, right? So for example, the crossing block from the breeding program, we screen it for a group of markers that we are very interested to know if that particular line has or has not the gene. And so here is an example for the how do we use adult all stage resistance. So this is Silvina's master thesis, where she was working on pyramiding 2 resistant genes, 3 actually, but he deployed them in combination of 2 for against stem rust. So the main thing in these genes is these 3 genes, they were introgressed from wild relatives, okay? And most of the time, these genes came with a linkage drug. So they are linked with some undesired DNA that we don't want it to, introgress in the breeding pipeline. So Ian Dundas group in Australia, they were working on shortening these translocations. And they came up with some lines that have the shortest translocation possible, but still having these genes. So we requested these lines, and we designed a scheme that we put 2 of these genes together, like 26 with 32 and 26 with 39 and 32 with 39. And then we started to do backcrossing and market-assisted selection with adapted lines to introgress these genes in adapted germplasm. So the thing is, we cannot put these lines directly into the breeding program, right? Because they are not adapted to Uruguay. Most of the time, they are not high yielding. They are very tall or very late mature. So we need to kind of set up like a genetic background to give these resources to the breeding program. So this is type of the work we are doing in our team. Another example here involving also stem rust SR2 gene, but also FHB1, that is a Fusarium head blight, a resistant gene. So in this case, these 2 genes are naturally linked in repulsion. So if you have one, you don't have the other. But in James Anderson group from University of Minnesota, they were just crossing lines, crossing until they found some lines, and they developed some lines that contain these 2 genes together in cis. So again, we requested the germplasm. And Miguel's thesis was about, kind of the same idea of Silvina's thesis. He choose 4 adapted INEA wheat cultivars, and he was doing a backcross market-assisted selection scheme to introgress this resistance into the adapted germplasm. So the final product, we get like backcross 3 F2 lines with the resistant genes in adapted background, and we send it to the crossing block to improve Fusarium head blight and stem rot resistance. The third example here is about the other plant-resistant genes, so the quantitative type of genes. And this was part of my master's, actually. And so what we did here is, because these genes are environment dependent, so they express differently in different environments, one of the things you need to know before deploying these genes in your breeding program is actually which of these genes is the most effective, right? So we knew that LR34, one of the genes, it was the most effective in Mexico, but we were not sure about Uruguayan conditions. And so we requested a population that was segregating for 3 of the genes to CIMMYT. They sent the seed and we put it in the field during 3 years. And we genotyped the lines with some specific markers for these genes. And we found many of the things that we already knew, like that these genes have a small effect, but they are additive. So if you put 2 or 3 or 4 of these genes together, the resistance levels are very good. And most of the time, these are pleiotropic, so they also confer resistance to other diseases. But what we found that was very interesting is that under Uruguayan condition, it was not LR34, it was LR68, the most effective gene. So we started to introgress the gene in the breeding pipeline, right? What we didn't knew about that time, this was back in 2015, is that LR68 was not pleiotropic against stripe rust. And so 2 years after this work, stripe rust started to be a problem. And so we took these same lines to the field in 2021 to screen specifically against stripe rust, and we found that LR68 does not have an effect controlling stripe rust. So this also gave us information about, okay, we need to stop a little bit on introgressing LR68, and we need to go to LR34 that is still effective in Uruguay for leaf rust, but also has an effect on the stripe rust. So this is a very valuable information for the breeding programs. Now I'll switch to the third major task on breeding for disease resistance, that is the development of resistant germplasm. And so everything we have talked about, from understanding the pathogen variability to exploring the host genetic diversity, ultimately it fits into this step. So to develop truly resistant cultivars, we need powerful genetic resources and mapping populations in order to understand what is the genetic basis you have on underlying in your program. So here I'm showing just a few examples on these populations. I'm showing the recombinant inbred line population that is crossing 2 parents, one that has the trait and the other one that doesn't have the trait that you are looking for, but is very good for other traits. Then you have nested association mapping populations and magic populations and the association panels. And of course, all of them has pros and cons. So I will move now to show you some examples using this type of population that we develop in our team. So here is an example for wheat stem rust, is the second chapter of Silvina's master thesis. So she took 2 lines from INEA that were adapted but they were susceptible to stem rust, and then an European line that was high yielding but susceptible to stripe rust. So she made 2 populations, one of 170 lines and the other one about 100 lines, and we put them in the field during 2 years. But most interestingly, we send it to Kenya. So we have Uruguayan phenotype and a Kenya phenotype. And so her main results were that SR24 and SR31 are effective in Uruguay. And she found out that some regions are effective in Uruguay and in Kenya. But she found also some regions that are very effective in Uruguay only. Okay, so these help us to pinpoint genomic regions. Okay, but at the end, what we are more interested is either to develop markers associated to these QTLs that we can then use to track this resistance across the program, or also to identify the wheat lines that are having these regions to use as parents in the breeding pipeline. And now our second example is focused on stripe rust. So it's another chapter of Venancio's PhD. And he was using an association panel to screen about 400 lines to screen against stripe rust. So he did field testing in 2021 and 2022, and he did GVS marker data, and he combined both, using statistics, and he run GWAS analysis, and he found 5 QTLs that are associated with the adult plant resistance, and a few of them are more likely novel. Okay, so he's now working on his publication and things, but again, we are able to design markers that specifically target these regions, and then we can, from these 400 lines, we can choose the lines that accumulates the more favorable QTLs, and we can use these lines to further do research or do more crossing in the breeding program. This other example is more like an integrative example. So it actually combines a lot of that we have been talking about. So the first I'll say is Ariel Silva master's thesis. He is from Paraguay. He joined last year our group, and so he will be doing mapping for Fusarium head blight resistant on a line that we have been using in the breeding program since a few years now. So we know that this line is resistant, but we don't know what is the source. I mean, it's not FHB1, it's not FHB2, there is not any of the genes that have been named for Fusarium head blight. So we develop a specific population, and it's about 200 reels. And we already have 2 years of field data. We are getting this year field data, and we got some GVS genotyping, and now we are just waiting to collect all the information to do the QTL mapping and find those regions that are responsible for the resistance on this specific line. And then the other chapter of his thesis is about like phenotyping some lines that have introgression from wild species. So we got some lines from the WGRC Center from Manhattan, Kansas, that these lines were like, they took Chinese spring and they crossed it with different wild relatives that were all good for Fusarium head blight. And now we requested these lines, and we have it here. We will put it in the field to screen against Fusarium head blight, with the aim to look for new sources or new diverse resistance that we don't have in the breeding pipeline. The third chapter of this work is about similar to what I've showed for stripe rust and FHB and SR2. Is kind of introgress the FHB7, that is another resistant gene that is kind of good for Fusarium head blight resistance, and we will be introgressing through a backcross marker assisted selection pipeline with the FHB1 lines. So we will take those lines that have the FHB1 SR2, and we will introgress the FHB7. So this kind of shows exactly the kind of integrative effort that is needed to build durable resistance into elite lines. And it's a great example of how modern breeding is leveraging multiple tools and knowledge, to put to introduce germplasm in the breeding pipelines. As a final example, and because Carla said that I am very interested in the wild relatives, I want to briefly highlight this project that we have been working since 2022. It's a project that is led by University of Maryland, Dr. Steve Warry's team. And so he put together a project that is like an international effort of different people from different places that is searching for useful genetic resistance from the Triticum monococcum accessions. So Triticum monococcum belongs to the A genome. So as many of you know, wheat is an hexaploid species that has 3 genomes, the A, the V, and the D. And so Triticum monococcum is A genome. And, you can, because most of the genetic resistance has been lost during the domestication and evolution, sometimes you need to go to the wilds to look for new resistance, and then you can introduce that resistance into bred wheat, okay? So in this sense, we put together an association panel of about 200 accessions, and we are phenotyping this against many different traits. So my job is focusing on diseases. So I did work on strabras, leafras, stembras, wheat blast, and also the wheat kernel mite. And so another team is doing all the genotyping. So they are doing whole genome sequencing and the pan genome and all these tools that are available now. So at the end, combining the phenotypic data with the genotypic data, we can run some very powerful analysis. And that at this type of resolution, we can actually just pinpoint the gene. So in this case, for example, I'm showing the wheat kernel mite phenotyping. So the wheat kernel mite is the vector of the wheat streak mosaic virus that is a very important viral disease for wheat in many parts of the world. And here we found a region on chromosome 5 from monococcum, and we found that it's an NBS-LLR gene. And now we can use these accessions that have this region to cross with wheat. But more interestingly, we can actually go to look for some synthetic wheats or some wheats that has these lines on the pedigree already, and we can search specifically if this region is already present on wheat. So this type of work is very exciting. I did some of this also during my PhD for wheat blast, and for the Tausches, that's the D parent of the bred wheat. So it's very interesting and very exciting to find new things that we can put together through the breeding pipeline. So before finishing, my talk was focused on breeding for disease resistance. So we have a few projects finishing in this year, and then others starting this year. We have the field-based phenotyping platform that we have been running since 2015 in India, that we score and screen germplasm for different diseases. And as I mentioned at the beginning, I just recently started to work with my team on breeding for physiological traits and for applied quantitative statistics. So in that sense, we have 2 new projects. One is it's a Fontagro project that we have with the University of Lleida in Spain and with the University of Rosario in Argentina, where my role would be specifically testing some isolines for photoperiod genes under different light conditions and temperature conditions, in order to evaluate if these genes have also pleiotropic effects on yield. Okay, so that would be a new student is starting soon to work in this project. And then we are also starting with a new project with CIMIT about the BNI trait. So BNI it stands for biological nitrification inhibition, and it's a trait that is not naturally present in wheat. It is present in other species, but not in wheat. And so a group in Japan, they found a wild relative species from wheat that has this ability to do BNI. And so they took this line and they crossed it with wheat, and they give these sources to CIMIT, and CIMIT put it on adapted wheat background. And now they got a funding to actually test a group of isolines, okay, so lines that has and does not have the gene in different parts of the world, and kind of to see if the BNI has an effect or not. It's like let's start from the beginning. Let's see if this works or not. If it doesn't work, we will forget about it. But if it works, it could make a huge difference, because, it will eventually, you need to fertilize less, and you will produce less gas emission and all that things related to nitrogen. Then for the quality traits, I want to mention one project that we are starting. We started actually last year with the Rural Development Agency from South Korea, that is about develop wheat that is hypoallergenic. So there is like in the Asian culture, there is a allergy related to some genes. And we found some mutant lines that doesn't have these genes, and we're just putting these in adapted germplasm in order to provide the South Korean community with this type of flower. And then we have a lot of collaboration with the School of Agronomy from the university here in Uruguay, doing a lot of applied quantitative statistics. So they are testing genomic selection and SPAR testing designs in the field, and how is better to allocate resources in the breeding program. Like if I need to test all the lines in all the environments, or if I need to test one line in one environment and a sister of that line in another environment, and then you use that relationship to predict the other one. So things like that is the type of research we are working right now. So before I wrap up, I want to take a moment to acknowledge everything that I have shared today is not the result. My job is the result of a collective work. I want to thank the incredible team of the Rust or the pre-reading. That is Richard, Fernando, and Noelia, some former students and former researchers, and the new students, and all the other scientists that are working at INEA, the Barley breeder and the wheat breeder, the pathology team, the molecular team, and also the national evaluation trial team, that they support us and they help us with this work. And also want to thank the funding agencies and all the collaboration that we have with other institutes around the world. So with this, thank you so much for listening.
Thank you, Paula. It's been amazing. Great presentation, and congratulations. And thank you so much. We have several questions already in the chat. I want also to thank Silvia Herman, who is in the audience too. I haven't seen Silvia for many years, but great to have you with us, Silvia. I'm going to start with some of the questions, okay? Ramesh I think it's from Pakistan, is asking, while multiple gene stacking, how will we make sure we are successful in pyramiding if we don't have molecular tools?
Yeah. So all of the examples I show about doing pyramiding genes, we use markers to track the gene. Sometimes, if we are talking about Rust and you have arrays that you know is virulent to that gene, a specific gene, you can use it to phenotype the line and kind of select the ones that you know that has or doesn't the gene. But in the examples I show, we always work with genes that we have molecular markers to track
Okay.
on the lines. Yes.
I think I lost your camera. I want to reactivate it. I
Oh,
don't
sure.
see you.
Sorry.
Okay, there you are. All right. Okay. Let me go down. Lucia Vignale is asking, is saying, "What a great talk, and how easy or difficult is to share plant material among countries?"
It's very difficult. So that depends a lot on the country. So some countries are very difficult, like Brazil, for example. Brazil is very difficult to share germplasm. And I need to say, is something positive that we have in Uruguay, and that is actually something that allows me to, introduce material all the time. Uruguay has a very good setup for phytosanitary permits and import permits and things like that. They are very smooth here. But yeah, it's not easy. So for example, now I have been trying to bring germplasm from Australia, and it's very difficult. Most of the time I need to request post-quarantine testing at the entry. And for that, I have a specific greenhouse space that we use to do the quarantine. And then people from the Seed Institute is coming to check, but yeah, it's not easy.
Okay. Thank you, Paula. Prince from Nigeria is saying, "Great presentation. And how do interactions between resistant genes LR34, 68, or SR2 influence the severity of wheat leaf rust in adult plants under varying conditions in Uruguay?"
Well, yeah. So that was part of my master's thesis. And so we found that the lines combining the three of them are the most resistant, of course. What we didn't check on those lines is yield, right? So most of the time, you will have a yield penalty. Not always, but you have some yield penalty because of combining a lot of these genes. One of the characteristics of these genes is they are linked to some morphological traits. So for example, LR34 is pleiotropic with the leaf tip necrosis. So the tip of the flag leaf will die. And that, of course, will have a yield penalty. But regarding the question about varying environmental conditions in Uruguay, so in Uruguay, I'll say we have more effect, like yearly effect rather than locations effect within the year, right? So we have a lot of year genotype by year interaction, let's say. And so what will happen with these genes is that some years they will express more, and some years they will express lower. So I remember last year, one of the release cultivars that we are promoting right now, it has the LR34. And last year, the expression of the leaf tip necrosis was very high. And I remember the farmers asking, like, what is this? What is happening with my plant? It's dying. And I was like, okay, it's not dying. It's something that is coming with that resistant gene. And they didn't like it, actually. They were asking to, okay, can we take it off? Can we remove it? I was like, well, I mean, if you want the resistant, you need to handle this. But yeah, the expression varies depending on the year for sure. And of course, the resistant expression as well, right?
Okay, Paula from Colombia is asking, what will be the breeding objective for your photoperiod work? Are you looking for a photoperiod insensitive lines? Do you have a golden standard for the response to that variable?
Yeah. So that's a good question. So So actually for photoperiod, we started very basic because we didn't know much about what was the combination of genes that explain the phenology on the breeding program. So we started like just genotyping a bunch of lines for the vernalization gene and photoperiod genes and earliness per se genes. And we choose 20 lines that they had different combinations. We put it in the field in 7 different sowing dates, kind of to study which combination of genes is the most accurate, which one is maximizing yield in Uruguay, right? And so after that, or because of that project that is a master thesis as well, we learned that the USDA germplasm bank, they had these isolines, photoperiod isolines. And so we requested the lines again, and we put we multiply the seed because most of the time they send you a small amount of grams. So we multiply the seed, and this year we are starting a new project that is testing these isolines, having like so for the photoperiod gene we have like A, B, and D genes, right? And each of them with the allele like sensitive or insensitive. So we have 3 pairs of 3 isolines. So one is sensitive, so it has the sensitive allele for both genes, B and D, because A gene doesn't have is monomorphic, doesn't have variability. Then we have lines that is insensitive for B, but sensitive for D, and opposite, right? The line that is insensitive for D and sensitive for B. And so we will put these lines in the field to test not only what is the effect on the long of the cycle, but also because it has been in the literature, you can read that the photoperiod is pleiotropic with yield, but we are not very certain about that. And to study pleiotropic effect, you need isolines. It's the only way to actually kind of test if that single gene is having effect on different traits. And so we are doing this year for the first time. Maybe in 2 years, if Carlos invites me again, I can show you.
That could be a specific topic for your next seminar.
Some results on this.
I'm gonna do, we have a lot of questions. I'm gonna jump to the last questions. So it's from Ignacio Terrile from Argentina.
Hi, Ignacio.
Yes, he said, your opinion about wheat blast in the southern countries, will it be important?
Okay.
And are you doing anything about it?
Yeah, that's a tricky question because he knows that I am working on that. So I actually had one slide about blast, but I just because of time I took it out. So what happened with blast is that it's endemic from South America, right? So it's endemic from Brazil in the 80s, and then it spread to Bolivia, Paraguay. It appeared in Argentina at some point, but it's not a main disease. But then in 2016, it moved through, it spread through continents, right? So it appeared in Zambia and in Bangladesh. So it started to gain a lot of attention, right? Not before, but after moving to Asia, in Africa, it started to be a main thing. And so it happened that I was doing some research during my PhD, and we were not sure about Uruguay, what was happening, right? So we were surrounded about countries having blasts, but not in Uruguay. So the hypothesis was, it's the climate conditions, it's a tropical disease, so we don't have the weather for the disease to develop. So in 2023, that we had we have environmental conditions in the northern states of Uruguay that were very similar to Brazil and Paraguay. And that year, the epidemics in those countries was very, very severe for blast. So we went to the northern state of Uruguay and we sampled with spikes with symptoms, and we found blast, right? Then we did all the searching about, the molecular part and the phenotypic part to actually, come up that, it is with blast, it's not another blast. And so we are about to publish the paper now. But I'll say for Argentina and Uruguay, that the region where we grow the wheat is not as tropical, as in the northern Argentina or northern Uruguay. I'll say right now it's not a problem. Of course, if we think about climate change, right, and rising temperatures, and if we think about the pathogen population and how these pathogens adapt to other conditions, like the striped rust, right, that it was like cooler, they are from cooler environments, but they adapted to warmer temperatures and they appear in South America. So kind of the same story. So I think we need to be careful. It's not a problem right now, but it might happen that it's in a few years start to be a problem. And the scary thing with this disease is that right now there is not a lot of resistance out there that you can use to deploy. So right now it's only a translocation, again, from a wild relative that is conferring resistance, but it's not always useful. So there is a lot of effort going on finding new resistance to actually try to see what is happening. But I'll say to Ignacio, for now, it's not a thing. But,
Okay.
something that we are doing is sending germplasm to Bolivia to evaluate against blast. So at least we start to know which cultivars that we grow in Uruguay are resistant or susceptible, things like that. We are already doing it.
Paula, I want to pick another question that is very provide some information on how INEA works, because there's a question, is INEA doing only academic research, or is the institute developing lines that are sold commercially? How does this pipeline work? I think it's worth, and this question comes from Julia Sherman.
Yeah. So INEA is the National Institute of Agricultural Research. So of course we the main thing under our research is the farmer, right? So try to give information to the farmers and try to give us with tools and things that they can use to better produce or better, perform in the agriculture. But INEA also is it's also breeding lines for I'll say cereal crops. So we have barley breeding program and wheat breeding program, rice, soybean, oat. We have also trees. We have some horticulture. So we have, I think it's 11 or 12 breeding programs.
Oranges.
Oh, yes, citrus.
Don't forget
strawberry, tomato. So we have a bunch of and also forages, right? Pastures. Festigloleum and others. And I'll say we are, it's very local, right? So we do breeding for the local conditions. It depends in each species how the thing is, for example, for wheat, we used to be the main providers of genetics. And since a few years, we started to have a lot of introduction from Europe. A lot of the European materials, they adapt very well to Uruguay. So now we need to compete to the European breeding programs. And we are actually losing a lot of
market share
area because they adapt very well and they are more high yielding. So INEA focuses a lot on disease resistance, right? Because you are the National Institute of Agriculture. You cannot release a line that, needs to apply like 4 or 5 times fungicide, right? But the European companies, they bring that material to the country. So that is I mean, there is a lot of discussion about it on this. But yeah, we are doing research, but we are also doing some breeding and releasing material. So it's a little bit
You
of
are, as an institution, I understand you license lines
yes.
to be tested or promoted in other countries. I do understand you have some agreements here in the US too.
Well, I'm not familiar with that. I mean, for wheat, we don't.
No, not for wheat, for other species, yeah. Mhm.
Oh yeah, for other species, yeah,
Mhm.
probably yes, yes.
Is it
Yeah,
okay, Paula? We have so many questions and I want to start wrapping up. It's been an excellent time. Is it okay if I post your email address for
oh
people to
sure, yeah,
contact you?
of course.
I'm gonna post Paula's email address in the chat. Please take note of that address and write to her. It's been a great presentation, Paula. Let me share my screen. Okay. Thank you so much, Paula.
Thank you
It's
for the
great.
invitation.
Yeah, and
And
thank you everybody for, yeah, go ahead, sorry.
no, that I just want to mention that we are always open to, collaboration and having visitors from other countries to do training or, like new students that want to start to do some their masters or their
Mhm. Yeah, and yes,
PhD. So they
people
are always
please
welcome to send me an
try to
email and we
pursue
can
that.
talk about it.
Paula lives in one of the most beautiful areas of our country, the Southwest.
Yes.
Thank you so much, people, from attending the seminar. It's been a pleasure, and I look forward to having you next Thursday at the same time, where we have Dr. Hela Rojo from Tunisia talking about cucumber, local melon, and germplasm and breeding at the same time. Thank you, Brandon and Tiago, for your help. It's been a great presentation, Paula. I hope you have a great weekend, and we'll stay
Thank
in
you so
touch.
much.
Okay?
Thank you.
Thank
Bye-bye.
you.
Thank you all.
Goodbye. Goodbye, everybody.
Bye. Goodbye.
Bye.